So on Monday, you are going to have a quiz. And one of the things that's going to be on the quiz is the genetics problems. And so I'm just going to show you how I would do a genetics problem using multiple choice format. Okay? So I'm looking at this disease that's called hemochromatosis. And it says that it is autosomal recessive. So what does autosomal mean? It's not on a sex chromosome, right? So it's on one of the 22 pairs besides the sex chromosome. So I don't have to worry about male or female on here, which we're actually going to do an example of, okay? And then if I'm looking at this, it says that it is autosomal recessive. So what I would do, and I do this every time I do our genetics problems, I put a little legend here. So I'm going to say H is the gene, right? So big H, is that normal or the disease? It is the normal, right? Because it's caused by a recessive allele. So this would be normal. And then the little h would be abnormal, right? So this would be the one that has the disease because hemochromatosis is a recessive condition. So it's caused by the recessive allele, okay? So the question is individual 2, 4. So I come down, this is generation 1, this is generation 2, so it'd be 1, 2, 3, 4, okay? So generation 2, 4, what is the genotype of generation 2, 4? Heterozygous. And the reason I know this is that I have to work at all the information that's provided in this pedigree. So this would be little h, right? Because they have an offspring with the syndrome, right? They have to be carriers. So a carrier is what is referred to as being heterozygous. Okay? Does this disease skip generations? Yes. Yes, skips the generations. Okay. Okay. So if we look at the Punnett square example, so it says from the previous, and that should be 2. Sorry, not should be 2. So from the previous question, individual 2, 4 and 2, 3 have another child. What is the probability that the child does not have the disease, right? So this is where I would do my Punnett square. Okay. So what's the probability that they do not have the disease? 75%. They do not have the disease. 25% is that they do have the disease. Okay. So that is kind of the format that you're going to get, especially for the final exam. The genetics problems are going to be kind of in this format. Okay. Are there any questions about that? Okay, so we were talking about pedigrees, and one of the things that we can designate in pedigrees is what is called consanguinity. So this literally means the coming together of what? What's sanguine mean? Does anybody know? Blood, right? So this is the coming together of blood. So this was very common in previous centuries where royal lineages, royal people believed that they had special types of blood, right? And so they wanted to keep their blood pure. And you can see this today, not in humans, but you can see it when we're talking about purebred dogs, for example, right? We keep it in the blood, we keep the lineage clean, right? So coming together. So if we look at an example of this in past times, we can see a pedigree showing Cleopatra's. I can't get it all on here, right? So this would be Cleopatra down here at the bottom, right? And so if we go back, so looking at this particular pedigree, the first time that we see consanguinity, and consanguinity is the double lines, what would be the relationship between these two individuals? They would be first cousins, right? So here you see first cousins having children, and then then first cousins having children. And then in this pedigree, we can even see, right, uncles and nieces, right? We can see brothers and sisters coming together to have children. So one of the reasons, and if we look at it in terms of other animals or even humans, but in general, when we talk about other animals and we talk about like, if you have purebred, say golden labs, sometimes they have specific genetic traits that are unique to them that might be detrimental, including things like juvenile cataracts, okay? So if we look at an example of why this might be significant, I'm just going to draw a little pedigree here, and I'm going to demonstrate first cousins. Okay, so those would be first cousins. And let's say that we get a genetic mutation, a really rare genetic mutation. So we could say that maybe it is Q. We'll use a Q. Okay. So this is a rare new. So this is a new rare mutation. Meaning that it's novel, that it is new, that it's never been seen before in this population. So this is the only person in this whole population that has this mutation. And you'll notice that the little q is the one that is the mutation, so that it is the recessive mutation. So it is recessive. Oops, recessive. Okay. These individuals have a child that has a disease caused by this mutation. The only way that you could have this mutation coming together into one individual is if their parents were genetically related to the person that originally had the mutation, right? Since the mutation only had one, it only arose once, right? We see inbreeding or consanguinity. So these individuals, right? Here would be Q, little q. This would be big Q, little q. These guys would be big Q, because it's not in the general population. And notice those guys are marrying in. Okay, so they would be carriers for this rare mutation. And so generally, this is why sometimes it's bad to have people that are genetically related coming together. But if you think about in past populations, if you think about some of the populations that gave rise to larger populations, when we talk about population genetics, sometimes we start out with very few individuals migrating to an area, and those are the individuals that give rise to the greater population. So consanguinity happened a lot in our past, right? Because we had a lot of small populations that moved into new areas and then expanded. And so I think something like, I think I read somewhere like there was originally like 1,000 people in Japan, and they gave rise to everybody who's in Japan now, except for those that immigrated in, right? So it's quite amazing that we have these founding populations. So when we look at things like cystic fibrosis, it might have been that this mutation arose once and then it spread through the population by this type of interbreeding, and why we get higher incidences of it. And so these guys are said to be hybrids, right? Hybrids, right? Because they are heterozygous, right? And then this one would not be a hybrid, right? It has 2 copies of the mutant allele, right? So when we think about organisms other than humans, when you think about dogs or maybe even plants, right? Hybrids are actually good because they can have a normal allele masking the presence of the recessive allele. But when you lose a lot of the hybridness, then you can get bad alleles coming together and forming and causing disease, right? Okay, are there any questions about that idea? Okay. Oops, what happened there?
I actually had one, Michelle.
Yes.
On the mother's side,
Yes.
how did the disease, because see the two big Qs,
Okay.
how did that happen? Because that's not actually part of it.
Okay, so these guys.
That's the normal.
So this is normal, right? So what I'm trying to point out here is the mutation only has to happen once, but if you have inbreeding, then you get this, and it's a reduced. They call it a reduced heterozygosity. Okay, yeah. So these guys married out. This one just happened to get this little Q from that parent and that big Q from that parent, and I know that because they had this child. But they could have equally have gotten a big Q. They had a 50% chance of being big Q, or big Q, little Q. But they just got the little Q. Okay. And we're going to talk about another example of this idea of consanguinity in a little bit. Okay. So we're going to talk today about sex-linked traits. And remember that this is on the sex chromosomes. So in humans, sex is determined by the sex chromosomes, and more specifically by the Y chromosome. In other organisms, that is not the case. So in your textbook, they talk a little about fruit flies. Fruit flies actually have a gene that determines the sex. And like some lizards and turtles, it actually depends upon their incubation temperature as to whether they become male or female. But in humans, it's a chromosomal cause. Okay? And so when you look at these sex chromosomes, and this is not a picture from your book, but this is a diagram, or excuse me, an actual image of the chromosomes. Now, are these chromosomes replicated or unreplicated? They are replicated, right? And so this obviously is the X chromosome, and this is the Y chromosome. So when we look at the X chromosome, it's much larger, and it has it's believed to have between 1,000 and 2,000 genes. Right? Whereas the Y chromosome has less than 50 genes. So much fewer genes. And so what this means is that when we talk about X-linked or sex-linked, we're generally talking about X-linked traits, right? Those that are caused by the X chromosome. So they've been able to map these genes onto the chromosomes. This is also not in your book. But this is the X chromosome, and it has certain dark staining regions and certain light staining regions, and they're just showing some of the genes. And remember that the SRY gene is actually on the Y chromosome, and that is what determines sex. We talked about that in lab. Okay, so if we look at the, go back. If we looked at the inheritance of the SRY gene, right? So we can talk about, this would be the female, right? And instead of a X for a cross, I'm just gonna put a little dot here. So it'd be like multiplication, right? With a little dot, and XY. So this is female, and this is male. So if we look at the female, she can only produce eggs with the X chromosome, and the male can produce sperm with the X chromosome or the Y chromosome. So coming together in our Punnett square, we would predict, on average, that half of the offspring born or produced would be male, and half of the offspring produced would be female, right? So it's the presence of the Y chromosome. So these offspring are male, and these offspring are female. But what they recently discovered is that this is not necessarily the case early on. So the Y chromosome is actually lighter. So this sperm with the Y chromosome is lighter, and it is faster. And so the number of male embryos that are conceived is greater than female. So the number of males conceived is greater than females. Okay? Because the Y chromosome is fast. The sperm with the Y chromosome is lighter and it's faster. Does that make sense to everybody? Okay.
Say that again.
So the Y chromosome has less genetic material, so it's actually believed to be lighter and faster than this sperm, and the fast sperm are the ones that get the egg, right? So we have more males being conceived than females, which is interesting. However, when we look at males, males have only one copy of all of the genes on the X chromosome. So if there's a bad gene on this X chromosome, the males are going to express it. So males have a higher early mortality, okay? So their males are more likely to have bad genes on the X chromosome expressed. Than females, right? And so they have a higher mortality. So for example, male embryos tend to be less viable and they're spontaneously aborted more frequently than female embryos. The other really interesting thing is it tends to depend, or it's believed to depend, upon the female's, the mother's environment. And so the sex of the child seems to depend Okay, so the environment, and I'm going to show you a little video, really quick. Oh, did I just erase that? I did. Okay. Increased mortality. What did I write? Sex. Okay, so let's say that there is the bad environment and the good environment. Okay, so is the mother really stressed out, right? Or is the mother really has lots of resources and is really mellow and, you know, confident in her ability to raise offspring? Okay, so I'm going to show you this little video, and this video talks, it's called Minute Earth, and it talks really fast. So you don't want to take notes while you're watching it, just kind of try to pay attention. And then we'll answer that question about good versus bad environment.
Assume the sex of their child comes down to a flip of the chromosomal coin, with an equal chance of having a boy or a girl. But in reality, the odds aren't even. For every 100 girls born, the world gains about 106 baby boys. Currently, that skewed sex ratio comes out to roughly 10 million more baby boys than girls born worldwide each year. There are some countries where human meddling stacks the odds even higher in favor of boys, but that doesn't explain the fixed odds everywhere else. The intrinsic boy-girl ratio is rigged by Mother Nature. In fact, it's even more rigged than birth rates show. Human conception results in about 150 male zygotes for every 100 females. But there's a rather tragic reason for this big biological boy bias early on. Male fetuses are much more likely to be miscarried or stillborn than female fetuses. And boys that do make it out of the womb suffer more fatal diseases, take more mortal risks, and fall prey to more violence than girls. So by the time kids grow up and reach baby-making age, the ratio of males to females is just about 1 to 1. But the likelihood of a boy even making it to birth is also influenced by his mom's living conditions during pregnancy. For example, when a massive famine struck China in the 1960s, the relative likelihood of having a son suddenly dropped until the famine ended. And male Americans born to billionaires seem to have higher than average odds of fathering sons. Somehow, female biology suppresses boy survival in the womb during tough times and boosts it when times are good. We've seen the same pattern in other mammals too. When resources are scarce, mothers give birth to fewer males than normal. When resources are plentiful, they bear more. The best explanation we have for this has to do with sex, the other kind. In biological terms, the whole goal of copulation is to reproduce, to pass down your genes to someone who will someday pass them on again. Female offspring are almost guaranteed to reproduce, famine or no famine, because male mammals are pretty much always willing to mate. Males, on the other hand, have to compete for mating privileges. A well-nurtured hunk has a good chance of mating with lots of females, while a male weakened by famine might not score at all. So male offspring are a bigger risk in general. At all stages, they're more likely to die. And even if they live, they might not reproduce. But when times are good, boys' potential to father lots and lots of babies make them a biological risk worth taking.
Okay.
Thanks to everyone who supports us on Subway. Without you, we literally couldn't.
Okay, so if the environment is good, what happens? Boys, right? If the environment is poor, we have more baby girls. Okay. Okay. So when we look at sex-linked diseases, we can talk about what is referred to as X-linked patterns. And we're going to specifically talk about X-linked recessive. Okay. So instead of autosomal, it's X-linked. So that means that it's caused by a gene on the X chromosome, and it's not dominant, it is recessive. And so an example of this is color blindness. And specifically the type of color blindness that allows a person to determine between red and green. So sometimes it's called red and green color blindness. So they aren't able to distinguish between those two. And so when we look at the gene, it is actually caused by a gene on the X chromosome. And so I'm going to put a C here with a little line over it, right? So that means that it is recessive. So this causes the trait. An X with a big C is normal. So now we have to pay attention to male and female individuals in the pedigree. So if we look at a female possibility of the genotypes, you could have this combination, you could have this combination, or you could have this combination. So it is incorrect to say that if it's X-linked recessive, that there would be no probability of a female getting the trait or the disease, because here is a female with the trait. Here is a female. These two are actually normal, right? This one's normal. Normal vision for both of these, but this one is a carrier. If we look at the male possible genotypes, we then still have X's, but we have only 2 possible genotypes. And so males can never be carriers for the disease. So if they have a copy of the bad allele, they will always express it. So that one with the little c is going to have the trait, and the other one is going to be normal. So I'm going to draw, or I'm going to write out a simple pedigree of a family with this trait. Now, one of the things you'll notice is that even in the small, and you have to look at a big, sample size, but tends to be more boys, right? So it only had boys drawn in here. Doesn't mean that there could not be a female, but it tends to show up more commonly in boys. Okay, so when I'm doing a sex-linked pedigree, I always start by putting my Y chromosomes in. And this just allows me to kind of remind myself that I have to include the sex chromosome, right? So I put the Y in. So the males that have color blindness are going to have a little c, right? This male does not have color blindness, so he would have a big C. Again, the grandfather up here would not have color blindness, so they have a big C as well. Okay, so what would this female have to be up here, this one right here, if in generation number 1, if she has this offspring? She has to be a carrier, right? Sometimes in pedigrees, what you'll see with carriers is that they will draw half the symbol in. And so that means that it is a carrier. And that means nothing. Okay. Okay, and the same thing has to be true for this individual. She would have to be a carrier. So she would be big C, little c. Now the question is, could you potentially have a female offspring that has color blindness from this cross, from these two parents? No. Because notice that she would have to get 2 copies of this chromosome, and the male always gives his female offspring the big C, right? So these children, the children have to, if they're female, they will never be colorblind, but 50% of the time they'll be carriers. That should be a big C. And 50% of the time they could be completely normal. That's a big C, big C, big C. So if I did my Punnett square, right, I would say if I have a female child, there is no chance that she would have color blindness. Now, what was the possibility that the male offspring, if they have another male offspring, what is the probability that that male offspring will have color blindness? 50%. And that's because it depends, since he gets his Y here, it depends upon whether or not he gets this X chromosome from his mother or this X chromosome from his mother. So 50% of the time, there's a 50% probability that if they're male offspring that they will have color blindness. So the thing about X-linked traits and sex-linked traits is it depends upon the sex. What's the possibility? Sometimes it's the same probability, but in this case, that it isn't, right? So the sex affects the probability of having the trait, what sex they are. Does that make sense to everybody? Okay. So another example that you could work in your lab, if you didn't, was the one that was on hemophilia, and specifically hemophilia A. So what does hemophilia mean? Blood loving, right? So hemophiliacs bleed, right? Their blood does not clot. And so this is actually due to a defective clotting protein in the blood that causes the blood to not clot. So they tend to die they used to tend to die relatively young, right? So we know in the past that there has actually been royal lineages. I think this is Queen Victoria from England. And this just shows it's passing down of hemophilia in this royal family. And if you look at it, you'll notice that the only ones that are completely colored in are boys. Now, why does this die out in this family? So why when you get down here, do you not see? Because none of the females are carriers. Yeah, but you don't even see it in the males. what, do you see none of those males are actually mating, right? Yep. So notice that if you have hemophilia and you're male, you have no possibility of passing that gene on to the next generation. So this just showed in the past, these people didn't live to reproduce, right? They died young, right? Yeah. So you could actually put a little line, you know, you could, if I was to do this, it'd probably be like, these guys died of hemophilia, right? Actually, this one didn't. That one actually is the exception. That's interesting. Yeah, that one did reproduce. Okay, I'm not sure how that happened. Must have had children very young, maybe. Okay. So now we're going to talk about kind of extensions to Mendel. So when Mendel was doing his work, remember that we either had dominant or recessive, right? But we actually have different degrees of dominance. And so we have a continuum. So I'm going to draw my little line of my continuum, right? And here we have complete dominance. So this is what we have been talking about. So this would be like Mendelian patterns, right? Tall is completely dominant over short. Green is completely dominant over yellow. When we talk about peas, that kind of stuff. Okay. At this other extreme, we have what is called codominance. And what that means is both alleles are equally expressed. And then, interestingly, in the middle, there's this big area here, this gray area, right, which is called incomplete dominance. Right? So this has to do with like how much of the recessive allele is expressed. So it kind of varies in some diseases. Right, so sometimes, and actually cystic fibrosis is actually is incomplete. We talked about it as a complete dominance trait, but actually has some incomplete characteristics. Okay, so an example of this in humans is familial hypercholesterolemia. So familial, when you see that, that tends to tell you it is genetic. It's a genetic disease. It runs in families. Hyper is too much cholesterol in the blood. We need cholesterol in the blood, but if cholesterol gets too high, then it starts to clog up the arteries, and it can lead to heart attacks, right? The arteries of the supply blood to the heart. It can also lead to strokes, right? So you clog up the vessels that are serving the brain. So when we look at this, we have two genes. FH1, which is the normal allele, and FH2, which is the abnormal allele. Most people have two copies of the normal. So I'll just put FH1, FH1. Right? So this is what is normal. FH1, FH2 is slightly elevated cholesterol levels. So you could have, say, for example, heart problems and circulatory problems maybe in your 30s and 40s. So problems starting in 30s and 40s. That's age, right? So your cholesterol levels are a little bit high, then you start to get clogged vessels, and you might have to go in and get surgery. Yes?
I'm sorry, but I can't tell which ones are two.
This is a 1 and this is a 2. Oh, it does, my 1s and 2s do look the same, don't they?
Yeah.
This is 1 and this is 2. This is 1 and 1.
1 and 1.
Yeah, 1 is up here. So why am I using 1 and 2s instead of just capital letter and lowercase letter? Because they have incomplete dominance, right? So I can't like use a capital letter and a lowercase letter because of the incomplete dominance. So I'll just put 1s. I'll not put my little funky little 1. So I'll just put 1 as a line, 1, 1. And this is 2. Okay. So then you have people with this, okay? And this causes problems. They're very elevated cholesterol levels. So that's elevated cholesterol, right? And then you have like teenagers. Oops, teenagers. So, you know, in your teens to 20s, you start to get people having these cardiovascular illnesses that they should not have. And this is actually, when you hear, sometimes when you hear about people, like a kid in class who's running around the field during PE and they all of a sudden have a heart attack, that could be due to this disease. And also like teenagers on the basketball court and all of a sudden they'll have a heart attack in a game, that can be due to this disease, right? So it just causes, you know, these heart attacks that can kill people early on, okay? So this is an example of incomplete dominance because, because the heterozygous individuals are different from the normal population and different from the people that are homozygous for the allele. So this is another sign that you have it, is that you can get cholesterol, so much cholesterol in your blood that it starts to get deposited in tissues, and so you get the deposition of cholesterol like in the elbows, right? That's one sign of that cholesterol coming out of the blood and coming into the tissues. So this just shows plasma concentrations of cholesterol. Okay. Now there's another example of incomplete dominance that we're going to look at in lab, so I'm not going to talk too much about it, but another example is called sickle cell anemia. So this is another example of incomplete dominance. Okay, so also Mendel was just working with traits that were caused by 1 or 2 alleles. But now we know that some of our genes have more than 2 alleles, right? So I'll put some genes have more than 2 alleles. Now, we only inherit 2 alleles, right? Because we're diploid, but in the population, there could be more than 2 alleles. So I'll put more than 2 alleles in the population. And an example of this is the ABO blood type. Notice I did not put any positive or negatives on there. So we're going to talk about that separately. No positive or negatives. So this is actually determined by what they label the gene I. I'm not sure why they use I, but that's the letter that they use for the gene. And there are 3 alleles. So there is big I with an A, big I with a big B, and little I. These are the 3 alleles. The A and the B are co-dominant, which means that they are equally expressed. The little I is recessive. So these 2 are co-dominant. This one is recessive. So when we look at the genotypes, remember that the genotypes are the combination of alleles. We could also talk about the phenotypes, which are the outer expression, which would be blood type. So all the possible combinations coming together, I could have this in a person. I could have this coming together in a person. Oops, dang it, I always do that. It's IAIB with a little b above. And little I, little I. So those are all the possibilities. And so now instead of like 2, whether you're tall or short, you could be type A, you could be type AB, you could be type B, or you could be type A. Okay. So this combination, these two give you the same combination, same phenotype. So this is type A. And what that means is that on your red blood cells, you have A antigens. Okay. So you have A antigens on red blood cells. So I'll put RBC, red blood cell. Okay. Type B, you have B antigens only, right? So B antigens. And then obviously AB is where you have both. Equally expressed, right? What about O? O is where you have a none. Okay, so you'd have no A or B antigens. So an antigen is something that generates an antibody. So an antigen is a protein that can cause the immune system to produce antibodies. Okay. So remember we talked about how proteins, plasmid membrane proteins, are important in cell recognition. This is an example of that. Okay. So which of these blood types do you think you could give to anybody and they would not produce antibodies? O, right? So O is the universal donor, right? Because it has neither A or B. So nobody, if you gave this to the person that was AB, they'd say, oh, that's great because it, you know, it's, well, actually, if you give a person with A, right, they would say, that's fine because there's no B antigens on it, okay? So which one do you think that you could, if you were this, that you could receive any? AB. So AB is the universal recipient. So this one's, can everybody see red? Okay, so this is universal recipient. That's AB. So if you're kind of a selfish person, you'd want to be AB, right? And then this is the universal donor. And luckily for universal donors, O is a very common, one of the most common types of blood, right? But they can give blood. So if you're O negative, for example, then you're really popular with the people that take blood. Okay. So you can actually use a Punnett square to calculate the probability that an offspring would be one type or the other. So let's look at an example of this. possibility. So let's say you had a person that was AB, and you crossed that with a person that was O. Okay, so that's my little X, that's my cross. So this was one parent, and this is the other. This is autosomal, it's not sex-linked, right? It's autosomal, it's not sex-linked. So this person, say this is the male, and this is the female. These would be the sperm, and these would be the eggs. The cool thing about this particular cross is that the offspring are like neither the parents, right? So they're not AB and they're not O. The only possibility is type A or type B in the offspring. Does that make sense to everybody? So RH has a different blood type. So we can talk about what is called the RH factor. It is a separate gene. So we can say we have two different alleles. Positive produces its dominant, right? And the protein is present. Interestingly, it's called RH because it was first discovered in rhesus monkeys, which have the same blood typing system. RH negative is recessive, right? So the protein is not present. Okay. So does somebody want me to tell me what their blood type is? Does anybody know what their blood type is? O negative. You're the popular one, right? So this is the phenotype. Now I can't tell that she's O negative by looking at her, right? But that's her blood type. It's not outer expression like you can see it, but it's outer expression because that's what's in the body, right? Not in the genome. Okay. So what is her genotype? So these two genes, right, form the genotype of O negative. Okay. Now, it's a little bit harder to tell some of them. Like if you told me, like I am A positive, right? So that's my phenotype. But if I were to look at the possible genotypes that an A positive could produce, I think there's 4 different possibilities. So I could be this. That would give me an R positive or an A positive. Positive. I could be that. Oops. Oh, sorry. Is that? Oh, yeah, that's right. RH positive, RH positive. Right. Or I could be this. That wasn't very logical, but. Okay. So if you look at all of those, right? A is completely dominant over the I, so I can be carrying the I and not know it. RH positive is completely dominant over the negative, so I can be carrying it and not knowing it. So all of these genotypes give me the exact same phenotype, right? Those are all the possible genotypes giving me a positive phenotype. Now I could know more if I knew what my parents were and what my offspring are. Right? I could maybe whittle it down and figure out what more specifically I am. Are there any questions about that idea? Okay.
So for like somebody, like my kids and I have the same blood type and their dad has a different blood type, so that would be.
I would have to know the blood types to figure out how that would work. Do you want to do a problem?
A and B's.
Okay, you figure it out and then bring it to class and we'll figure it out. Okay. Oops. Okay. So we now have some more examples of how genes interact to form the phenotype. And this is specifically referred to as epistasis. So this is called two or more genes interacting. To influence a single trait. Okay. So eye color has been taught for a long time as being really simple with just B's, right? One gene, little big B, little b. But it is actually much more complicated than that. And so when we look at eye color, it is not a single gene. In fact, there might be like 16 or maybe even more different genes interacting. But the example that I'm going to give today is how you could explain parents who are blue eyes could potentially have an offspring that is brown-eyed, right? So you're like, oh, that doesn't make sense from what I learned in grade school or middle school or high school, right? But it's a lot more color or a lot more complicated than that, okay? So I'm just going to use brown and blue eyes. So we're going to totally ignore green. Green is actually the rarest eye color, and it actually has to do with the distribution of melanin in the eyes. So we have a gene called O, and this produces melanin. And melanin is the brown pigment in the eye, and specifically in the iris. Okay. It is also the pigment that gives us our skin color and also our hair color is melanin. Melanin is our pigment, okay, that we use in our body. So we also have another gene that is called gene H. And this is necessary for the production of melanin. So it's necessary for the production of melanin. It might be an enzyme, for example. But gene O and gene H both interact with one another to produce either brown or blue eyes. So we have to know both of them to kind of understand this, okay? So in most of our population, we have dominant copies of the O, right? And dominant copies of the H. This is brown eyes. Sometimes we see individuals that have this combination, and this is blue eyes. So when we look at the inheritance of this, something really is interesting about blue eyes. And so if we look at kind of what we've been able to discover about blue eyes recently, so this is actually 2008, but what they recently were able to discover is that this genetic mutation that gave rise to blue eyes actually arose in the population 6,000 to 10,000 years ago And it was by a single ancestor. So it says blue-eyed humans have a single common ancestor. Okay, so if we go back to what this single common ancestor looked like, right? So they were able to kind of reconstruct where they think blue eyes originated from and what population. And it was in the Mediterranean region, right? And this is the kind of a representation of what this person probably looked like. Okay. So they took a DNA sample and they found out through the DNA analysis that this person has blue eyes. So this could be our blue-eyed common ancestor, okay? Okay, so if we look at the genetics of it, we could potentially have a person with this combination, mating with a person of this combination, right? And this could produce a person with blue eyes. And so actually, I misspoke at the beginning. These are brown-eyed parents, right? This is blue-eyed offspring. In this model, the simple model, there's no way of having blue-eyed offspring produce brown-eyed, or blue-eyed parents producing brown-eyed offspring. But there is a way to do it. It's a little more complicated than this one.
Okay, did you say there's no way?
There is a way, but I'm not going to explain it.
Well, my granddaughter's does that.
Oh, really?
Yes.
Okay,
She
well,
has
we can.
bright blue eyes, and my son and the mother of my grandbaby both have very, very dark brown eyes.
Okay, well, I'll try to explain it then. Question? You want to know the answer too?
Well, because I have a bigger brother. I have two brothers actually who have blue eyes, and one of them has blue hazel. And my stepdad has brown eyes, and my mom has green brown hazel.
Well, that's way too much information for me to put in my brain to answer any question. Okay.
Sounds complicated.
Okay.
Maybe. How do hazel eyes work? Because I have hazel.
Hazel eyes has to do with where the melanin is deposited in the iris. So the iris kind of has these ridges and grooves in it. It's kind of like mathless, right? And so it has to do with the reflection of like where that is deposited. And so an eye color can actually change by light coming in and being reflected back out. So sometimes your eyes might look more green, and some days they might look more blue, and that has to do with the refraction of light coming out of your face. Okay. Okay. So let's just show this as an example of a blue-eyed offspring. So how does this, how can you say that we came from a single ancestor that had blue eyes? How does that make sense to you? How could you have a single ancestor with blue eyes? This is something that we talked about at the very beginning of class today. No, we talked about, what was our first topic? Consanguinity, right? So if you have a mutation arising in one individual, right? So this mutation arose here. This was the first mutation. Right? So they can kind of trace it back and figure out when that mutation happened. And that mutation might be so specific that everybody that has blue eyes has this specific mutation. And so they say, oh, well, it must have come from, because the probability that the gene would mutate in that specific way is very rare, right? So it was a specific mutation. And so these two individuals, right, had to have been related and had to have had at some point a common ancestor back in time that gave rise to the blue eye. Okay. So I want to show you, just as a very ending of today, another little video that talks about consanguinity. And it also, interestingly, talks about our interaction with the atoms in our body and the atoms in the atmosphere. And it is called We Are All Related. Okay, so he starts out and he talks about genetics, right? And he talks about the genetics, and then he gets down to the subcellular level, and he talks about atoms. And you have to understand this, you have to realize that carbon, there's only so much carbon on the planet, and so we take carbon up by eating, and we put it into our bodies, and then we breathe it back out. And so the carbon that exists in me was the carbon that was found at the beginning of the planet, right? So we're constantly recycling molecules and atoms through our body. Okay, this is another fast one.
Hey, this is Michael here, and you guys are my family. Scientifically speaking, geneticists tell us that every single person on Earth is at the least 50th cousins with everybody else on Earth. It kind of has to be that way. Think of it like this. You have 2 biological parents and 4 biological grandparents and 8 biological great-grandparents. It keeps doubling. Well, if you follow your family tree all the way back to the time of the ancient Romans, 64 generations ago, that tree would need 1 quintillion separate individuals, which is a number that is larger than the number of humans who have ever existed, ever. So what does that mean? Well, I think Bill Bryson put it best when he said, you couldn't be here without a little incest. Actually, quite a lot of incest. Nothing ridiculous, mind you, but if you go far enough back in your family tree, you're going to find that quite frequently, people who were distantly related had kids. In fact, if you are currently dating or married to somebody who is from your own country and is of your own ethnicity, there's a 1 in 5 chance that the 2 of you share a common family member fewer than 10 generations ago. Furthermore, because our society is becoming more and more mobile, our most recent common ancestor is becoming more recent. Mathematical models are now showing us that for everybody, every human on Earth right now, our most recent common ancestor probably lived only 2,000 to 4,000 years ago. A recent study, it's a great one, you should read it, found that there was a person alive as early as 300 BC who is related to every single person alive on Earth today. Their study also found that that person most likely lived in Taiwan, near a busy port city, so their descendants could quickly spread around the world. Now, all that said, you only receive half of your genetic information from your mother and half from your father. And as you go up your tree, it's quite possible that distant ancestors had literally no contribution to your genome. So, who are you? I've said this before, but because bacteria are so much smaller than the average human body cell, your body contains more bacteria than it contains its own cells. And forget about trying to remember every individual cell and bacteria in your body. They're constantly dying and being replaced. Even cells like brain cells, which seem to live for a very long time, still take in new nutrients and expel waste. Meaning that within about every 5 years, every single atom that used to make up your body has been exchanged. Do you remember what you were like 5 years ago? Well, the atoms that make up your body, they sure don't. This is a little existentially scary, but it leads to a very good question. How many atoms are inside my body to begin with? How much is a billion atoms? Well, here's one way to think about this. If you were to count one number greater every second, it would take you 12 days to count to 1,000,000. If you wanted to count to 1,000,000,000, that would take you 32 years. So 1,000,000,000 is a very large number, but not when it comes to atoms. Atoms are so inconceivably small that even if you had 1,000,000,000, say, sodium atoms, their entire weight would only be 38 femtograms. Each individual cell in your body, too small to be even seen by the eye, contains 100 times more atoms than there are stars in the Milky Way galaxy. Multiply that by the number of cells that make up what we call our cells, and each human body contains more atoms than there are stars in the known universe. Numbers that large lead to some pretty mind-blowing truths. For instance, this one mentioned by Bill Bryson. Because our bodies contain so many atoms, and because we so vigorously recycle them, statistically speaking, about 1 billion of the atoms inside your body used to belong to Shakespeare. 1 billion of your atoms used to belong to Beethoven. 1 billion of the atoms in your body right now, as you sit and watch this, used to be inside the body of Buddha. You could also think of it this way. 1,000 years from now, when humans first land on a planet outside of our solar system, that first person to step onto the surface of the alien planet, their body will contain 1 billion atoms that are in you right now. And as always, thanks for watching.
Okay. Any questions? Oh. So every 5 years, you have entirely new set of atoms in your body.
I always thought that was every set that your body.
That's your cells.
Those are your cells. Like your skeleton is rebuilt every about 7 years.
Because they always say too that like you didn't like something. Yeah. 7 years ago, you should try it again because.
Oh, is that like the 7-year itch?
Well, no.
Ow. It's like every 7 years.
Because the cells in your head, where do you grow?
I never heard about that.
So every 5 years, what is it?
You have an entirely new set of atoms in your body. So all the carbon, oxygen, nitrogen, phosphorus, sulfur, all of that has been exchanged.
So my blood type is A positive, and then dad.
So you're A positive, Dad,
B positive. And then both kids are definitely B positive.
so you are definitely. Let me figure that out. Okay. So you are A positive,
Yes,
and the father is B positive,
correct.
and the children are both B positive.
Yes.
Okay. So I know from this that you have to be this. He could be, so if he is this, then the children have to be this. I don't know about him. He could be something else. So the children are this, and then you just know that the children, since you guys are both positive, you don't know about RH. So it could be this, or so the only information that gave me is that you have to be this, and the children have to be that.
All right.
And I have to think about the blue-eyed offspring.
Yeah, because it caused like issues because everybody was like, how did that happen?
Well, what if you got?
And they took DNA tests and definitely they are the mama and the daddy.
Right.
Because, and it's weird because it's not just a little bit, she has like crystal blue, crazy blue eyes. And they both have like the darkest brown eyes you can possibly get.
So what was?
That was, I didn't quite understand.
Oh, this would be it. So that's the grandparents. So The little o means that they're blue-eyed here because of that gene, and this one's blue-eyed because of that gene. And so then they could produce an offspring that is this. Which is brown. So these are blues, and that one's brown.
So you're saying.
So if you have recessive, there's another gene here, so these two interact, right? So if you have two copies of the recessive O, right? So, and two copies of the recessive H, those are two different ways to get blue eyes. Right? And so that would be the parents, and then they would produce a child that has a big O from that parent, a little O from that parent, a big H from that parent, and a little H from that parent, and they would have brown eyes. That makes sense?
And the brown
And
eye
that
produce
is
the
the
blue eye.
So yeah, blue-eyed parents, right? Wasn't that what we're asking?
Brown eye parents, blue eye child.
Or was it the opposite?
My son and significant other both have really dark brown eyes.
Oh, brown eyes, okay.
My granddaughter has really blue eyes.
Okay, so this is the example, ignore that, right? So this is the brown-eyed parents, this is the blue-eyed offspring. That's what they had to have. So they were each carriers for that little H, and they produced an offspring that had blue eyes.
And so, and some of the grandparents do have blue eyes. So that's what made it possible. Because the other.
Yeah, they inherited the recessive allele from their grandparents.
Because his dad, my ex-husband, blue eyes.
Right.
And on her side, her mother had blue eyes.
I mean, I understand.
So they, each of the parent of my grandkids had a blue and a
And
brown.
she's got blue eyes even after she's grown up.
Yeah, well, she's kindergarten,
Okay.
first grade.
Because, you know, babies oftentimes have blue eyes.
No, this is like,
This is
she
real
will have blue eyes. She's, I mean, there's nothing back. It's
Yeah.
like, bam, big blue eyes.
Yep.
Bluer than anybody in like 4 generations. Yeah. You've
So
seen
then,
Kelly.
yeah,
Yeah.
so then, I have this down.